So welcome everyone. Welcome to Nutrient Cycling in Crop Systems, Impacts of the Web on Soil Food Web on Nutrient Availability by or with Alexa Smychkovich. And today, as we like to start off each webinar and each presentation, we use this map to remember that we're occupying land that has been inhabited long before European colonization. And this is a great resource where, as we all work towards our equity and inclusion goals, we can, remember to acknowledge and learn more about the folks who lived here for a long time before European colonization and are still here today. We can click through this interactive map and find more information about the folks living in our regions. As well, we want to make sure that you enjoy your presentation. So to do that, we'll keep ourselves on mute. And at times we may come off of mute to ask questions and make comments. Just remember to put yourself back on mute to preserve sound quality. As well, we invite you all to use the chat feature and put all your questions into the chat. And then we can bring them up as necessary during the presentation or at the end during the question and answer period. And this session is being recorded. We thank again our sponsors who help make our education and advocacy work possible. We encourage you to support many of these folks yourselves and to thank them for their support of NOFA. As well, I hope you've been able to check out the NOFA Summer Auction. It's live online. You can go and see some of the really great items that are up for bid and see if you can get one of those things yourself. We also encourage you to spend some time in the virtual vendors meeting room where you get to learn more about our vendors and all their great work, and we really thank them for being here. It is my great pleasure to introduce Alexa, who is a PhD candidate in the Plant and Soil Sciences program at Stockbridge School of Agriculture. Her research is focused on enhancing soil health and increasing the yield and quality of vegetable crops using cover crops and arbuscular mycorrhizae fungi.
Thank you.
Thank you.
Thanks for the introduction, and I'll just share my screen and get started. All right. So can everybody see that and hear me okay? All right.
Yes.
So just wanted to say thank you for everybody for coming to this presentation. So as Doug said, I'm a graduate student in the Plant and Soil Sciences Department at UMass Amherst in Stockbridge. And so some of my thesis work focuses on nutrient cycling and soil food web interactions. And so I'm by no means an expert on these topics, but as I've been getting more involved in this research and doing a lot of reading and putting some of this research, adding some of this research into my own work, I've just found it so interesting, and that I just thought it would be so great to be able to share some of the topics that I found especially useful to me to kind of contextualize some of these ideas. So here they are. I'll just go ahead and get started. So first and foremost, what is the soil food web? So the soil food web documents the relationships between the complex community of organisms and the environmental processes that exist in the soil. And this is important because organisms within the soil food web actually regulate a lot of the ecological functions that we rely on to grow crops, to sequester carbon, to do a whole lot of things. So the organisms within the soil food web, first and foremost, they redistribute minerals through space and time. So what does that actually mean? Well, the space could be pretty easy to understand. So you have these nutrients that are becoming available, and the soil organisms are moving them around the soil. They also redistribute them through time, because as they take up nutrients and then have them in their bodies, and then they die, those nutrients then become available once more. And so now you've had this redistribution through time as well. Additionally, these organisms directly influence soil fertility through both mineralization and immobilization of important plant nutrients, and I'll talk more about that later on. They decompose organic matter, which is one of their most important functions in agricultural systems. They modify soil structure, so very important for aggregate formation and aggregate stability. They regulate pest populations through competition with other, like for instance, fungal pathogens or bacterial pathogens. They sequester carbon by bringing it into the soil. And lastly, they degrade pollutants. So some bacteria are actually able to degrade pollutants that have entered the soil. So let me talk a little bit about what the soil habitat looks like. So picture here is an aggregate. So it's basically a really small structure in the soil made up of soil particles. And the point of this picture, this is taken from NRCS, is just to show all the different things that can be happening in one aggregate of soil. So of course you have these big pores, so you know that soil has macropores and micropores, and those store oxygen and water, which are two things that microbes really need in order to metabolize. And so in one of these aggregates, you have all sorts of things interacting all at once. So here you have plant root hairs, decomposing plant cells, you have mycorrhizal hyphae, bacterial colonies, nematodes. They're all existing in the same exact habitat, sort of just, well, they have their own little unique niches, but they're all existing in that same sort of area and interacting with each other all the time. And they're also really sensitive to different things. So organisms are influenced by, first and foremost, environmental conditions. So those are things like soil texture, the porosity, the pH of the soil. Most microorganisms like a near neutral pH. Of course, there are some that will survive in an acidic situation or in a basic situation. But for the most part, if you have a neutral pH, you can have the greatest diversity of soil microorganisms. Temperature, this is an easy one to think about. And if you think about the seasons, obviously there's not a lot of activity in the winter. If you live in New England, it's too cold. But then as it starts warming up and you get to summer, you have a lot of microbial activity. Of course, soil moisture, microbes need access to water in order to do their thing, and availability to oxygen availability. Soil organisms are also really sensitive to inputs. So one easy thing to think about is pesticides and fertilizers. So that really changes the way that soil microbes are going to behave. But of course, inputs can also be things like crops. You have different crops growing in the soil, and they're going to attract different microbes while they're alive. And then once they die and decompose, their residues are also going to attract different groups of microorganisms. And lastly, disturbance. So soil microorganisms form these little habitats in the soil. And when you disturb them, you're messing up their flow and you're breaking up these habitats. So of course, tillage is the big one, but it's important to remember that things like synthetic fertilizers are also considered to be a disturbance in the soil food web. And soil fauna. So that's things that like, voles or insects. Those things are moving around, and they're moving around, decomposing residue, and they're also causing disturbance on the soil surface. And one thing that's really important to remember is that soil food webs and processes are always in flux. So it's not like you have a soil food web and it's just going to be behaving the same exact way all the time. And it's because soil organisms are so sensitive to all of these different things. So for instance, in the winter, you're going to have different things happening than you have in the summer. Even if it just rains, then you have one part of your field that's flooded, you have anaerobic conditions, that's going to be behaving differently than a part of your field where it's already dried up and you start to have oxygen being reintroduced into the soil. So all of this stuff is always changing. So let's get down to the basics and talk about carbon in the soil food web. So carbon is important in the soil food web, first and foremost, because it drives the soil food web. Soil microbes rely on carbon from organic substrates for energy, growth, and reproduction. The decomposition of organic residues is a really important part of the carbon cycle, and it's a major function of soil microorganisms, especially in agricultural systems, because in agricultural systems, most of the carbon is coming from decomposition of residues. And so one basic thing to remember is that the more carbon you put into a soil food web, the more microbial activity you're going to have and the more trophic interchanges. So basically, the more carbon, the bigger your soil food web. So here's a quick picture of what the soil food web actually looks like. So you'll notice that you have all of these different levels. I'll go over those in a minute. But let's start from the beginning. and figure out how does carbon enter the soil food web in the first place. So there's several ways that carbon can enter the soil food web. The first is through living plants. So living plants provide carbon in several ways. The most directly, the living roots provide carbon to plant parasitic nematodes and mycorrhizal fungi. And this is a direct way of providing carbon to the soil food web, because these plant parasitic nematodes will actively feed on plant roots, which will actually cause a lot of crop damage, but they'll be assimilating carbon into their biomass. Mycorrhizal fungi is a little bit different. This is a symbiotic relationship in which plant roots are able to form these associations with the specific group of fungi that enters the plant root and forms structures that are able to exchange nutrients with the plant. And so the plant gives the mycorrhizae carbon, and the mycorrhizae is able to provide the plant with greater access to nutrients that the roots cannot access. So if you picture plant roots, yes, they proliferate into the soil, but it takes a lot of energy for the plant to create new roots. And they actually can't penetrate into all of the different levels of the soil and access all of the nutrients they need to access. Mycorrhizal hyphae, however, can do that. So it'll look for nutrients in the soil and send them back up to the plant roots in exchange for carbon. And now that carbon is going to not only go into creating more hyphae for the mycorrhizae and mycelium to explore the soil, but some of it's also going to be released into the soil by, first of all, hyphae turning over and decomposing, and also just through various exudates. So another way that living plants can actually release carbon into the atmosphere is through a form of organic matter that in the form of plant root exudates. So plants create lots of photosynthesis to help them perform their various functions. But a lot of these photosynthetes, they actually release into the soil. So approximately 30% of plant photosynthetes are exuded into what's called the rhizosphere. So the rhizosphere is a kind of like an area around plant roots. You can think of it kind of like a film. And it's the area where plants exude these photosynthetes, which are really carbon heavy, and feed the microbes that are living in the rhizosphere. So there's actually the most amount of microbial activity around the rhizosphere. And it's good for the plants to have these microbial interactions. And so there's this sort of interface between the plant roots and the microbes in that way. Also, as roots, there's fine root hair turnover, or as roots die or parts of the roots slough off, those also decompose and release carbon into the soil food web. And lastly, residues of dead plants enter the soil food web via decomposition. Of course, if you apply amendments like manure, those also will enter the soil food web via decomposition and also provide a certain amount of carbon to the soil food web. So here's that photo of the soil food web once again. So this is from NRCS, which is actually a really good resource if you want to learn more about this and you're looking for things. They have a lot of answers and a lot of pages that are really describe a lot of what's going on in the food web. And so the first thing that you might notice is that there's these different levels here. And these are trophic levels. You have the producers, which we just talked about a little bit. And those are eaten by this first level, trophic level here, which is then consumed by this trophic level, and so on and so forth. And so one thing to remember is that organisms within a trophic level have the same nutritional relationship to the primary source of energy, which is the producers or the plants, and share the same function in the food web. So every single one of these trophic groups has the same relationship to the producers and shares a similar function in the food web. Another thing to keep in mind is that these producers are giving a lot of carbon to the food web. And so you say this is the amount of carbon that the food web starts with. With each trophic interchange, the carbon accumulated by each individual organism decreases because some of that carbon is going to be respired, some of it's going to go into the biomass. So as you move on through the food web, less and less carbon is going to be available to these higher trophic groups. And so if you remember a few slides back, we talked about the more carbon you have, the bigger the size of your food web. And that's because you'll have more carbon for all of these trophic interchanges to occur. So just to sort of reiterate about the producers, so there's the living plants that have the rhizosphere, which supports a great amount of microbial activity. And another cool thing is that these roots provide access to a great range of microorganism populations, because as they grow down into the soil, now they're exuding these photosynthates on all sorts of different depths. And so that's providing food for microbes, not just near the soil surface, but further down in the soil profile. Another thing about living plants is because they're constantly photosynthesizing and they're constantly releasing these exudates, this is a really consistent supply of nutrients. So you always have this carbon going out into the soil. When you have organic matter in the form of manure or plant residues, most of it's located at the soil surface. And the residues are actually distributed by microarthropods like springtails or earthworms. And you have these transient nutrient flushes because you have to have the correct environmental conditions for decomposition to occur. And so say if it's really dry, the microbes aren't active, nothing's really happening, but then you have a rain and you have good conditions for decomposition. And all of a sudden there's these flushes of microbial activity. So these two inputs are a little bit different. So let's move on from the producers to the primary consumers in the soil food web. So again, directly, we first have plant parasitic nematodes, which parasitize the roots of plants. And here I have two images. There's actually two ways that plant parasitic nematodes can parasitize roots. So the first, and this is kind of hard to see, but this is a gall on a root. And so there's actually a nematode living inside of that. And so some nematodes will actually enter the root and create kind of like it's called a nurse cell. And they'll actually program the plant to send a lot of photosynthase to that one cell so that they can feed on the photosynthase. And so this is the root knot nematode melodogyne that causes these galls. Another way, and those are pretty host specific. There's also nematodes that are kind of more like grazers. And so they'll move through the roots and they'll graze on some of the roots on the outside. They'll never enter the root, and then they'll move on to the next plant. And those are not very host specific. So they'll attack a wide range of plants. And that's one way, a direct way that carbon gets into the soil food web through these primary consumers. There's also fungi and bacteria. So bacteria will assimilate plant root exudates, so those photosynthates being sent out by the roots. But fungi and bacteria will also decompose organic matter. And they're able to really be the first to sort of attack and decompose residues because they have a lot of nutritional versatility and fast metabolism. So there's a lot of fungi and bacteria that can kind of jump at these residues and start decomposing the sugars right away. So here's a little bit more about bacteria and fungi that serve as primary consumers. So bacteria in the soil, so bacteria in general are very abundant. They're microscopic, single-celled organisms, and they have really rapid generation times. So they can respond to inputs pretty quickly. And there's a couple of different groups of bacteria that live in the soil. There's actually a little bit more than this, but these are just the ones I'm going to focus on because they seem like the most important for some of the topics I'll be talking about today. So you have decomposers. Those feed on root exudates from living plants or plant residues, and they convert nutrients from their inaccessible organic form to an inorganic form that now plants that are actively growing can take up and also other microbes can use. That's called mineralization, and I'll talk more about that in a little bit. They also can keep nutrients in the root zone via immobilization. Again, I'll talk more about this. But when bacteria actually consume these nutrients and they hold them in their biomass, those nutrients are now unable to leach out of the soil. So they're kind of being held in the root zone. And so once that bacteria dies and those nutrients become available again, plants can now uptake this nutrient. It's not lost. Actinomycetes are decomposer fungi that actually, there's a photo of an actinomycete here that actually is a bacteria, but it behaves a lot like a fungus in that it forms these hyphal-like structures, and it actually degrades some of the same components of plants that fungi do during decomposition. Another type of bacteria that exists in the soil, which I'm probably familiar with, is nitrogen fixing bacteria. And this is also, you can also get inoculants with nitrogen fixing bacteria like rhizobia. So nitrogen fixing bacteria form symbiotic relationships with leguminous plants. And so the way that they do that is that these nitrogen fixing bacteria can actually break the double bond that exists in atmospheric nitrogen and make it available for plants to use. So plants can't do that. So the nitrogen fixing bacteria make these associations with plant roots where they form a little nodule and the plant gives some of its carbon to the nitrogen fixing bacteria in exchange for nitrogen access. You also have nitrifying bacteria. Those convert ammonium, which is a form of nitrogen that plants actually can uptake, to nitrate, a form of nitrogen that's also readily uptaken by plants. And this happens really fast in the soil. So you'll mostly see nitrate in soils. You're not going to see a lot of ammonium because this is happening really fast. This nitrifying bacteria is hard at work converting ammonium to nitrate. You also have denitrifying bacteria that converts nitrate to dinitrogen, so atmospheric nitrogen, or nitrous oxide gas. And that happens in anaerobic conditions. So waterlogged, flooded conditions, you'll see this denitrification occur. And of course, you have plant pathogens, which cause crop damage. So you also have soil fungi existing in the root zone. So fungi are microscopic cells that grow in thread-like structures called hyphae. And when the hyphae forms a mass, it's called mycelium. Sometimes you can the hyphae will also form these ropes, and those are called rhizomorphs. So if you've ever seen like honey mushrooms in the woods, and you see these white strands that look kind of like hyphal ropes, those are called rhizomorphs, and those are just strands of this mycelium. But the different types of fungi that I'm going to talk a little bit about today are decomposers or saprophytic fungi. So those are responsible for the depolymerization of more complex substrates and residues, so things like cellulose and lignin. They keep nutrients in the root zone the same way bacteria might via immobilization. So once they've consumed them before the population has turned over, those nutrients are in use. They're not exiting the root zone. They're also an important in the formation of humus, which is the portion of soil organic matter that is resistant to degradation. So it's really stable. And of course, it does degrade over time, but we're talking centuries rather than, months or years. Then you have mycorrhizal fungi. So I've already talked a little bit about mycorrhizal fungi and some of the benefits they have to plants. They form these symbiotic relationships that help the roots access water and nutrient. One of the big nutrients that they help the plant access is phosphorus, but they can also help with absorption of micronutrients and nitrogen. Mycorrhizal fungi also play a really important role in aggregate formation. So there's research that has come out that shows that mycorrhizal fungi, which produce a substance called glomalin, which is really just kind of like a sticky glue, is really, really important for the formation of soil aggregates, for having these soil particles stick together and form really good soil structure. And of course, there's plant pathogenic fungi, which cause crop damage and are responsible for major economic losses worldwide annually. Let's move on from the primary producers to the, to the secondary, this primary consumers to the secondary consumers. So the secondary consumers are going to eat the primary consumers. And so you have bigger organisms such as arthropods, which consume fungi, protozoa, which consume bacteria, and free living nematodes, which actually can consume both. And when I say free living nematodes, all of that really means is that they're not plant parasitic. So plant parasitic nematodes need living roots to feed. Free living nematodes feed on things like fungi, bacteria, other nematodes, or they can even have omnivorous diets. So of these soil fauna, or these higher level predators, you have microarthropods. Those are found on or near the soil surface in plant litter. So those are right at the top there. Some of examples of those are springtails, which feed on bacteria, fungi, algae, but also decaying plant material. So they'll actually be able to shred a little bit. We call them shredders. They'll go through the decaying plant material and they'll sort of munch them up a little bit so that other organisms can get to them. Mites, which feed on fungi, nematodes, again, decomposing plant residues, and small arthropods. And for springtails, if you can't picture what one looks like and you've ever been walking in the woods in the snow and you see these little things, they look like fleas that are kind of jumping up, those are springtails. Next, there's earthworms. And so these terms are not that important, but this is something that I personally thought was really interesting, is that not all earthworms are actually the same and they don't actually have the same habitat and they behave differently. So you have in soils epigeic worms, and that means upon the earth. And those live at the soil litter interface. So again, they're actually not going into the soil at all, and they eat decomposing litter. So they're eating decomposing litter, they're excreting it, they're grazing. Then you have anisic earthworms that come out of the earth. And these are cool because they actually have this surface. They actually feed at the soil litter interface, but they also create these tunnels vertical tunnels into the soil. And that's cool for a few reasons. First, they can modify the soil structure and they can actually aerate the soil in these ways and introduce oxygen, which microbes need for their metabolism. But they also can accelerate decomposition by dragging soil surface litter down into the soil where microbes can actually access it. And then you have endogeic worms, which create these horizontal continuous burrows under the soil surface, and they never really come up. And those feed on mineral soil. And then, of course, you have free-living nematodes. So for people who are unfamiliar with nematodes, they're roundworms, and they're found in every ecosystem on Earth. And there's really large nematodes that you can see with the naked eye, but for the ones that we're studying in the soil, for the most part, you need to look through a microscope. They're small enough to occupy soil pores and aggregates. And so here's actually a photo of a free-living nematode that I took this spring that's found in some of the fields that I'm working in. That's a fungal feeder. And how I know that that's a fungal feeder is that you can actually tell by the esophageal region of nematodes what their feeding habit is. So here's a diagram of the heads of nematodes. So this is right here. If you look, this is the top of the nematode. Here's the tail. So this is the esophageal region. And you can actually see that the top, there's kind of head looking thing, looks very different. And so this on the left is a bacterial feeder. You see this little cup, it's called a stoma. Bacteria, bacteria will enter the stoma and it'll absorb them that way. This is a fungal feeder. So you can see it has a very different feeding habit. It has this little needle thing. It's called a stylet, and it actually will penetrate hyphae to draw out contents. This is a plant parasitic nematode. So it has a stylet as well, but it's a lot stronger because it needs to penetrate plant roots. Here you have a carnivorous nematode. So this is actually a tooth. It has a little tooth that it'll use to eat other nematodes. And here is an omnivore. So these are really common in soil habitats, and this can actually eat a whole lot of different things. This is called a spear. It's kind of a jack of all trades. And lastly, you have your higher level predators. And so these are your arthropods. Also, you have nematodes in this category, these predaceous nematodes that eat other nematodes. And so these improve the efficiency of decomposition and influence the microbial community size by shredding, so redistributing the soil litter, and grazing. And what grazing means is that while they're eating other soil organisms, they're excreting maybe some nutrients, or they're kind of accelerating this nutrient turnover. So let's get into residue decomposition. So this is how carbon enters the soil food web. This is the primary way that it will in agricultural systems, is through residue decomposition. So what happens when residues decompose? Well, first and foremost, you have carbon going into microbial biomass and being respired as CO2. You also have humus formation, which is the stable amount of part of soil organic matter that is resistant to degradation. And you have mineralization. So mineralization is the conversion of organic compounds into inorganic compounds. So the inorganic compounds are things that the plant can take up. When they're in their inorganic form, it cannot take up those compounds. And that's a general term. So molecules can be immobilized or mineralized by soil organism metabolism, and they're released either through respiration or through excretion into the soil. And so ammonium and nitrate, the two inorganic forms, can become plant available through this mineralization process, and that in this way they'll contribute to soil fertility. So when I'm talking about residues, for the most part, I'm talking about cover crops, because that's the way that you're going to get the most residues into your fields. So cover crops are crops that are planted for their ecological benefits rather than for economic harvest. And there's a lot of reasons to plant cover crops. First and foremost, they recycle nutrients, including nitrogen, by taking them up and releasing them for successor crops while they're decomposing. And that prevents leaching. So if you have nitrogen in your soil and you're going into the winter, that nitrogen is going to leach out over those winter months. But if you have a fall planted cover crop, it's going to use up that nitrogen. And then in the spring, the residues will re-release it. So you're keeping those nutrients in the system rather than losing them by just planting a cover crop rather than keeping soil bare. They also prevent erosion, first by holding topsoil with their root systems, but also reducing the velocity of runoff. So you have a rain event or wind, it's moving across a field or across soil. If you have a lot of plants there, it's going to have much less velocity. And so you're going to reduce the amount of topsoil that you lose. Of course, they also feed soil microbes via their root exudates while they're alive and fine root turnover. And then once they're dead through residue decomposition, they also sequester carbon in the soil by bringing atmospheric carbon down into the soil. And they can alleviate compaction. There's some cover crops that do this better than others. Of course, like the one that comes to mind is forage radish or tillage radish, because it alleviates compaction so much. But in general, if you have these roots always in the soil creating these channels, you're going to have a much less compacted soil than if there's nothing there. And so what factors influence residue decomposition in agricultural systems? So first, the type of residue, and that means not just the species, but also, is it above ground? Is it the aerial part, or is it the roots? If you are not tilling your system and you have, or for instance, you have a cover crop that's been winter killed, those roots are still under the ground. And so they're going to decompose too. And they're going to decompose at a little bit of a different rate than the residue at the soil surface. Of course, the residue quantity, and that's influenced by the species. Some cover crops are going to produce a lot of biomass, like oats. Some, like forage radish, aren't going to have a lot of biomass at the surface at all, but they will have biomass under the soil surface. The time of planting, so how long are you going to have these cover crops in the ground? Time of planting and time of termination. Of course, residue management. So if you till the residues under, then they're going to usually decompose much more quickly, because you're doing a lot of the work for the microbes. You're putting the residues closer in contact with microbes. Of course, you're also destroying microbial habitat. So it's kind of a double-edged sword there. In no-till systems, you might have slower decomposition, but a lot of times that's a really good thing, because it's more perfectly timed with the growth of actively growing plants. Environmental factors, so soil moisture, pH, nutrient status, aeration, and temperature, those all influence microbial activity. So naturally, they're going to influence decomposition. The magnitude of the microbial community, so how big is your soil food web already? And residue composition. And this is what I'll spend some more time talking about today, because this really influences microbial communities. And it's something that I think is pretty important. So that includes the carbon to nitrogen ratio and the composition in terms of lignin, hemicellulose, and cellulose. So composition, so lignin, hemicellulose, cellulose, influences plant residue decay. So there's groups of organic compounds that make up plant tissues, and those, like I've mentioned, are lignin, cellulose, hemicellulose. And And those are selectively attacked by soil microbes that produce the enzymes necessary to degrade the polymers. So not all soil microbes can degrade all parts of a plant, basically. The microbes that can degrade a plant stem are not the same microbes that are going to be degrading, like, for instance, plant leaves. And that's because microbes produce different enzymes, and they need these different enzymes to degrade the polymers found in these structures. So simple substrates, for instance, sugar, can be attacked by a lot of different types of microbes. A lot of microbes, a variety of bacteria and fungi, are really ready to and able to assimilate these really simple substrates, and they'll attack them right away, and they'll feed in competition with each other, and their populations will boom. The more complex compounds like cellulose and lignin will selectively attract these groups of microbes that can break the substrates down using their enzymes. And as a general rule, the more resistant some substrate is to decomposition, the less microbes there are in the soil that have the enzymes that are needed to break it down. So celluloses are the most abundant structural polysaccharides in plants, and they degrade really, really rapidly via depolymerization. And there's a lot of soil fungi and bacteria that can attack and degrade cellulose. So those go really fast. If you've ever seen a residue decomposing, you might notice that things like leaves decompose really fast, and then you're left with things like stems. Hemicelluloses are also abundant. They comprise about 30% of plant residues, and they degrade just about as rapidly as celluloses. Lignin, on the other hand, is prominent in woody tissue. So if you think about things like straw or plant stems or trees, you have a lot of lignin in those woody tissues. And it's really resistant to decay, and it can decompose very slowly, mostly via fungal decomposition and with actinomycetes. So remember, those are the bacteria that kind of behave like fungi. So here are the stages of decomposition, just to get a little bit of an idea of what's actually happening. So in the first stage, you have your easily mineralized components coming from, these are plant residues. I don't know how clear that photo is, but these are plant residues, dead plants. So first you have the easily mineralized components like sugar of residues that are utilized over days or weeks. And from those, 50% of the carbon is going into fresh microbial biomass, and approximately 50% is respired. So you have half of that carbon going to fresh microbial biomass. Next, you have cellulose and other carbohydrates in the plants. So again, things like the leaves are being attacked and decomposed. And out of the carbon from those compounds, about half of it's being respired, and about half of it's going into this new generation of microbial biomass. This old generation of microbial biomass that exists, it's now kind of turning over. So a lot of the microbes are dying, and they're becoming food for this new generation. So you have a little bit of a bigger population here. Next, you have the third stage of decomposition. So now you have your lignin decomposing and the synthesis of humic material. So this can actually take a long time, sometimes over a year. Again, depending on all of those things that we've talked about, like environmental conditions. But you have this lignin decomposing. And so the organisms that decompose the lignin are a little bit less efficient. So now a little bit more of the carbon is going to be released as carbon dioxide, and about a third of it is going to go into this new generation of microbial biomass. And again, same as before, the generation of microbial biomass that you had is turning over and feeding this new generation. And of course, you have a small amount of the decomposing residues are going to go into humic production. So humus is physically and chemically stabilized and becomes resistant to decomposition. And there are different theories on how humus is actually formed. So there are theories that say it comes from the decomposition of lignin. Some people say it comes from from just basically byproducts of microbial decomposition. But there are different theories, but the most important thing is that it's formed and actually has a lot of benefits for the soil. It can increase, so it obviously increases the amount of soil organic matter found in the soil. It has a slow release of nutrients because it decomposes over a very long period of time, talking centuries. And also it can increase the cation exchange capacity, builds soil structure. So there's a lot of benefits to having a lot of humus in your soil. So now that we've talked a little bit about the carbon transformations during decomposition, I want to touch on the nitrogen transformations and availability. So the nitrogen in plant residues, as I've mentioned before, exist in an organic form that cannot be accessed by growing plants. So just because you have residues, if you plant a plant into residues and they're not decomposed yet, it's likely that that plant is not going to absorb anything from those residues because it cannot access the nitrogen that is in those residues. It's in a completely inaccessible form. Plant roots take up inorganic nitrogen in the form of ammonium, so NH4 plus, or nitrate, NO3. And how do we get there? How do we get from this organic form to this inorganic form? The process is called ammonification. And so that is just a process of mineralization. So this is done by soil microbes. Mineralization is a general term. Ammonification really just is very specific to nitrogen. And so it's the process by which plant unavailable nitrogen is converted to plant available inorganic nitrogen, which is ammonium and nitrate. And so microorganisms do this. But what else can happen? Immobilization. So some of the carbon from these, the nitrogen from these residues is not released to plants in an inorganic form, but instead is assimilated by the microbes because they also need nitrogen to produce DNA and amino acids. So when they're using up the nitrogen and converting it to their biomass, assimilating it, it becomes unavailable to plants and to other microbes. So the nitrogen is still there in the soil, but it's not available for plants to use. And this is called nitrogen immobilization. So I'm sure everyone has heard of carbon to nitrogen ratios of residues and that they're very important for nutrient cycling. And when you're choosing cover crops, you're choosing certain carbon to nitrogen ratios. So why does that matter and what does that mean is what I'm hopefully going to answer in the next few slides. So here on the left, you have a table of the most common, or not the most common, I'll say some common materials that could be put into gardens. So you have straw at the top here and microbes at the very bottom. And so you'll have the carbon to nitrogen ratios that with the lowest ones at the bottom, highest ones at the top. So one thing you might notice is at the top where you have all of these really high carbon to nitrogen ratios, you'll see straw, different types of straw, rye straw, wheat straw, oat straw. And closer to the bottom, you'll have manure, different types of manure, alfalfa, hairy vetch, a legume, and soil microbes at the very bottom. So one thing that a lot of these materials have in common with that have the high carbon to nitrogen ratio is that they have a high lignin content. So they have a lot of stems or woody residue. And the residues with the low carbon to nitrogen ratios have high proportion of cellulose and less lignin. And so you have, for your high carbon to nitrogen ratios, sorry, got a little distracted. The general rule is that if you have a carbon to nitrogen ratio of over 20:1, that you're gonna have nitrogen immobilization in your soil. If you have it less than 20:1, then it's likely that there's gonna be nitrogen provided in your soil. And here you might notice there's a little, right in the middle of this table, there's a section called the ideal microbial diet. And so that's 24:1. And what that means is that at 24:1, you're gonna neither have extra nitrogen in your soil, but you're also not gonna have immobilization. Things are just gonna be functioning the way that they're functioning already. So let me talk a little bit more in depth about what these ratios actually mean. So for every, basically what a ratio is that for every 24, for instance, for the microbial diet, for every 24 units of carbon, you have 1 unit of nitrogen. And so all of these ratios are going to obviously be a little bit different, but the main idea is that for a certain amount of carbon, you have a certain amount of units of nitrogen. So let's say that the ideal ratio for the microbial diet is 24:1, right? So for every 24 units of carbon that microorganisms need, they only need 1 unit of nitrogen. So let's look at a residue, for instance, red clover, that has a low carbon to nitrogen ratio, such as 10:1. So for every 24 units of carbon that microorganisms need to build biomass, they receive 2.4 units of nitrogen, but they only need 1. So now they have an extra 1.4 units that they can release into the soil via excretions. And that extra nitrogen can be taken up by actively growing plants. So you've created a surplus of nitrogen. What happens when you have a high carbon to nitrogen ratio? Well, similar story. For every 24 units of carbon the microorganisms need, they need 1 unit of nitrogen. But now, for every 24 units they consume, they only receive 0.3 units of nitrogen. And so where are they going to get the rest of the nitrogen? From the soil. And that means that they're going to compete with actively growing plants for the nitrogen that's available. However, microorganisms are much more efficient than plants at finding that nitrogen and using it. So they're going to basically outcompete the plant almost every time. The other thing to remember is you remember that high carbon inputs mean a greater, larger food web. So if you have these really high carbon inputs, you have a huge soil food web, but now it needs to sustain itself. And so it needs that nitrogen. So you have more organisms to feed. And so now you have nitrogen immobilization. So this kind of doesn't stop at the level of litter decomposition. This kind of moves through the soil food web. So there are some other ways that nitrogen can be excreted in the soil food web during these trophic interchanges. So the first is through grazing. So even though in the table before it said that the microbial carbon to nitrogen ratio is 8:1, that was an average. So it can be anywhere from 5:1 to around 12:1. So if you have organisms with different carbon to nitrogen ratios that are going to eat each other, you're also going to have release of nitrogen. So for example, if you have a nematode that has a carbon to nitrogen ratio of 6:1, it eats bacteria that has a carbon to nitrogen ratio of 5:1. So for every 6 units of bacterial biomass that the nematode consumes, it's going to have an extra 0.2 units of nitrogen that it's going to excrete into the soil. Also, if you think back to the decomposition slide where we look at the various stages, you'll notice that microorganisms are going to use 50% of the carbon that they consume to build biomass, and they're going to respire about half of it. So when you have organisms that have the same carbon to nitrogen ratio, for example, 5:1, in order to build 1 unit of biomass, that an organism needs to consume double the amount of carbon. But if it's getting double the amount of carbon, it's also getting double the amount of nitrogen, and it doesn't need that. So it'll be able to excrete the excess unit of nitrogen into the soil. So I'm going to talk a little bit about the management of crop residues and why these carbon to nitrogen ratios are important and why they're useful to think about during when you're thinking about the soil food web. So synchrony is really important between residue decomposition, nitrogen mineralization, and the nitrogen uptake of actively growing crops is really, really important for effective nutrient management. So if you have things with high carbon to nitrogen ratios, you might have nitrogen immobilization, and you might think, okay, that means reduced nitrogen availability to successor crops. If you have low carbon to nitrogen ratios, that seems like a really good thing because now you're encouraging nitrogen mineralization and you can contribute a lot of nitrogen to the soil. But the problem is if you don't have something that's actively taking up nitrogen at that time, that nitrogen's not going to hang around in the soil. It's going to leach really readily down. And by the time whatever you're growing is there to capture it, it's going to be gone. So cover crop mixtures are a really good way to kind of balance the carbon to nitrogen ratio. But of course, you can do this in other ways. So a good way to experiment it with it is if you have something with a high carbon to nitrogen ratio, you might want to add something that has a lower one, because the idea is to really optimize the nitrogen release over a period of time. Depending on what you're growing and the days to maturity and how much the vegetative growth period, you're going to want to have nitrogen available to the crop for a longer period of time. It can't be a year from now, so it can't have immobilized nitrogen, but you also don't want it all being released right away and then not being able to feed the actively growing crop throughout the growing season. So this is kind of like, I guess like a art and a science, as they say, but it's really interesting and something that I think people have fun experimenting with is these mixtures. And so just to reiterate, factors that favor rapid residue decomposition and delayed residue decomposition. And this is just to kind of plan for management, I guess. Rapid decomposition is favored by residues with low lignin content, so less woody stems, just visually, while slow decomposition will have high lignin content. Rapid decomposition will occur if you have adequate amounts of soil nitrogen or low carbon to nitrogen ratios that can support the soil microorganisms. Slow mineralization will occur if you have insufficient amounts of soil nitrogen or really high carbon to nitrogen ratios. Again, if you have favorable conditions for your microorganisms, so you have adequate soil moisture and oxygen, warm soil temperatures, near neutral pH, those are going to favor decomposition and healthy microbial activity. If you have cold soil, think about the winter or pH extremes, you're not going to have as much. In the cold, you're not going to have microbial activity. pH extremes, you're going to have some microbial activity, but again, not a large variety of microbial activity. And then dry conditions, again, not enough moisture for microbial activity to occur. Or if you have really flooded conditions, you might have anaerobic conditions, so there's not enough oxygen in the soil, it's all filled with water. Then you have conditions that favor fermentation. So again, conditions are very important. So kind of the conclusions here. Soil organic matter is really important. It provides energy and nutrients to support a large and diverse microbial community over a long period of time. It increases soil cation exchange capacity, water holding capacity, and improves soil structure and aggregation. Soil management systems such as no-till, reduced till, the use of cover crops, and other amendments can increase soil organic matter over time and support soil biology necessary for on-farm nutrient retention and cycling. So again, also some of these management practices are just disturbing the soil less. So not only are you giving energy to your soil food web, but you're also allowing it to kind of go through these trophic interchanges over a period of time without disturbing it and sort of breaking these linkages. And again, synchrony between residue decomposition and active crop growth is crucial for recycling nutrients and reducing off-farm inputs, and can be better achieved by experimenting with different ratios or amendments to your soil. So that's all I'm going to say about this general topic. I'm also going to talk a little bit about my own research and how I'm using some of these concepts in the field and in the lab to actually study these ideas and why I got into this in the first place. So I'll talk a little bit about that now, unless do people have questions that they wanted to ask first?
And if you do have any questions, you're welcome to take yourself off mute and ask those questions or put them in the chat.
All right. Okay. I'll keep going then. So,
sorry.
Oh, go ahead.
Alexa, there was a question in the chat from the beginning from earlier on, and she, Lynn was wondering whether microbes die in the winter or just go dormant.
Oh, they just go dormant. So that's a great question. Yeah. So microbial communities will survive the winter, but they won't be metabolically active. All right. So now I'll just move on to this part of the presentation, which I just want to talk a little bit about some of the work I'm doing at UMass. So this is my thesis work, and it's funded by the Massachusetts Department of Agricultural Resources and by Stockbridge. So the purpose of this project is to enhance the sustainability of garlic production through the use of cover crops. And basically, in general, usually garlic is planted in October, either into black plastic or into bare ground and then mulched. And so the purpose of this system is that, or the idea behind the system is that we plant cover crops in September and we let them grow until they're winter killed in December. So they produce enough biomass to be, I think, significant. And the garlic is planted into the rows of standing cover crops in October. So as it sprouts, again, when it's planted, garlic will produce roots in the fall, but it won't compete with the cover crops. And then it'll sprout through the residues in the spring. And hopefully at that point, the residues will start to decompose and maybe feed some of the, supply some of the garlic with nitrogen. So the benefits of the system is that the living roots of the cover crops will provide soil microbes with food and with energy until the crops are winter killed for a longer period of time. I know some people will plant cover crops and then terminate them and then plant garlic, which is great. The idea behind this is that you have a longer period of time with living roots in the soil. The decomposing cover crop residues recycle nutrients to the growing garlic. They also support soil biology. And the cover crops build soil health by preventing erosion and improving soil structure. So here's just some photos of the field experiment. This is conducted at the South Deerfield Crop and Animal Research and Education Farm. So here is the planting of the garlic happening in October. So you can see the cover crops are actively growing, and I'm just making furrows with a hoe and then planting by hand. Here, there's a couple of photos of the actual planting. So you'll see the garlic's just going pretty close to the residues right in between the rows. This is forage radish. This is oat. The 3 different cover crops we used were forage radish, oat, and field pea. We also used a mixture and then planted into bare ground and mulched just to have kind of a control. So here's the residues in January. So these have been winter killed. You can see the oat residues have a lot of biomass. Those are the straw looking ones here. And here's garlic sprouting through the oat residues and growing through winter pea residues. It's a little bit bigger at this point. And so how do we track decomposition in the field in order to figure out what the benefits of the system are? So what I did was in November, after the cover crops were done growing, but before they were winter killed, I took a sample of biomass. So I knew exactly how much biomass I was working with. I also dug up the roots because, again, since these are going to be winter killed, the roots are also going to be decomposing. And I took a measurement of the root biomass as well. And then I made these litter bags. And so these are just made out of window screen mesh. They're just stapled together. And they're filled with a known amount of fresh cover crop residue. And I also took a ratio between stem and leaf because, again, there's this different composition of the cover crops and they're going to decompose at different times, and I wanted them all to be pretty similar. And so this is what they look like. You have this fresh residue in the mesh bags, and I placed a lot of these on the soil surface. I also did this with the roots. I dug them up, weighed them, buried them back under the ground, and so they stayed there all winter. And then as the soil temperatures started to increase and microbes become active, I started to harvest these bags. So I took a couple of bags every 2 weeks, and I looked at how much biomass was left, changes in carbon to nitrogen ratio, and the nitrogen content in the residues. And that's to create a curve so I could figure out how much nitrogen is being released from these various residues and how quickly are they actually decomposing. So simultaneously, I wanted to look at the changes in the microbial activity and the soil food web. So the way that I did that is that every 2 weeks when I took up the litter bags, I also took soil samples from each cover crop plot, and I did some laboratory analysis. So first I measured microbial respiration. So here's a picture of how I did that here. And basically what you do is you take a known amount of soil and you give it a little bit of food source and you measure the carbon dioxide that's respired, and you can correlate how much biomass there is, microbial biomass there is in that amount of soil. I also tracked changes in food web using nematodes as ecological indicators. And so nematodes can be used as these bioindicators, and they'll actually track changes in a soil food web as resource availability shifts during decomposition. So that's really what I'm interested in, is as these residues decompose, what's happening in the soil food web and where are these flushes of microbial activity. And the reason that nematodes can be used as biological indicators is, first and foremost, they're present at all levels of the soil food web. So you may have noticed that there's nematodes in every single trophic group. They also have a really short generation time. They respond really readily to inputs. So if you have, for instance, something decomposing and you have the bacteria attacking it, the population of bacterial feeding nematodes is going to explode. So you'll be able to see really quickly this response, nematode response to what's happening, and understand what the soil food web is doing. And of course, I've mentioned this before, but the morphological features can provide information about the food source, and they're pretty easy to identify. Also, they're easy to extract. All you really need to extract nematodes is water. We speed that process up in the lab using a centrifuge and sugar, but you really only need water, and a lot of labs only use water to extract nematodes. So it's really efficient and it's easy. And so that's another reason why they're convenient for this type of analysis. So last thing, we're also going to quantify the benefits of the system to garlic yield and quality. So the garlic yield is going to be measured from various cover crop treatments. Here's the garlic that I've harvested this year, curing. So I don't have data on this yet. This is my first year, so the data will be ready next year. Also, we'll look at the nitrogen use efficiency, which will track which tracks crop response to available nitrogen. So we'll use that to understand how much nitrogen was contributed to the garlic from every cover crop treatment. And we'll look at the nutritional quality of the garlic bulbs. So look at the micronutrients that are in the bulbs, and we'll do that in the lab as well. So this is just image credits. I've shared this presentation, so if you, I didn't take all of those photos. If you're interested in some of them, here's where to find them. And of course, you can contact me if you want to learn more about the project or have any other questions or want to just talk. Here's my email. And with that, I'll wrap it up.
Great. Thank you so much. I'm going to jump.
I got a question, if it's all right.
Oh, of course.
Sorry, my video is not working. Thank you for your presentation. I'm curious. So we have a field that, was recently flooded with all the rains, and we planted our broccoli and cabbage there. It's still super mucky, and there's like this green algae growing. We cultivated it, but it seems like it probably just totally lost all oxygen. So I'm curious, do you have any approaches to what to do in that situation? And should I like broad fork it while the plants are in there, or to try to aerate it, or
yeah, I guess yeah, I don't have a lot of good suggestions. Again, a lot of the stuff is kind of long term. Like for instance, for building soil structure, that's a long-term thing. But yeah, I guess like my intuition would be, yeah, maybe introducing some air into the soil. So yeah, like maybe aerating it with a fork or something like that might be helpful, just because at this point your soil is completely waterlogged. So not only do the microbes not have any oxygen, but also plant roots, which the plants also don't have any oxygen. So you have these completely anaerobic conditions. And so if you can introduce oxygen in any way, I think that would definitely help. But again, I don't know if you're in the Northeast, but yeah, it seems like we have had so much rain, and we're going to get so much more rain that,
yeah,
just from like a practical standpoint, I don't know how much, just aerating would help or not, hopefully it does.
Thanks. Yeah, I'm in Concord, Mass. So.
Oh yeah.
We got a lot of rain.
I know, and I think we might get some more tonight.
Yeah. Thank you.
Thanks for your question. Are there any other questions? Sorry, I really sped through that. I think I tend to be a fast talker once I get going.
Alexa, I thought everything was really informative and interesting. So yeah, thank you for going through it in the depth that you did. I was wondering regarding your research, like you said that you're using forage radish, oat, and field pea for your cover crops. Are those all mixed together, or do you have them separated?
Both. So I have them all as monoculture, and then I have a mixture of the three.
Okay. Are you finding a result either in your soil or your garlic as a result of one way versus another?
So I haven't done the data analysis on it yet. So we just harvested we harvested the garlic at the beginning of July before all the rains. But yeah, it's hard to say. I can only say anecdotally. There are some things that I've noticed. For instance, like the garlic was sprouting for some reason sooner in the oat plots than it was in any of the other plots. One thing I will say that was weird this year, and I don't imagine it'll happen twice in a row, is that our oat residues didn't fully winter kill. So the oat actually survived the winter. And so the garlic was growing with the oats. That being said, when we harvested the garlic, there were really no noticeable yield differences. So that was one thing I was worried about. And it also didn't look like it had any, again, visual deficiency. Even though it was growing really in between oats and not residues. I talked to a farmer who grows his garlic this way, and he said that his oat did die. So it must have just been a kind of like freak occurrence because of maybe a warm winter. But yeah, no, I haven't analyzed a lot of the data. So next year I'll definitely have more to say that's not anecdotal. So yeah.
Thank you. I have another kind of more general question as well, if there aren't any immediate questions, but I learned a little bit about biochar yesterday morning during one of the presentations, one of the workshops. And I was wondering, are you familiar with biochar at all? And have you considered it at all in terms of a soil amendment, or do you have any opinion on it?
No opinion. I'm familiar with biochar, but I haven't, I've learned a lot about its benefits. I don't have any direct experience with it. And I haven't really learned about it in the context of like the soil food web or how it's metabolized in the soil. So, it sounds like it has a lot of promise. But again, I can't comment too much because I don't have direct experience with it.
I understand. One of the things that I was most interested in, it was both the potential for as a long-term storage mechanism, but then also I was also a little concerned about the role it would have on the C-to-N ratio. So it seems like it has really good potential, but also a few concerns in that regard as well.
Yeah, yeah. Well, thank you. Does anyone else have any questions?
Feel free to put those into the chat if you do.
And also feel free to email me if you do think of anything, or you have any suggestions for me. If you think of something interesting in my research and you have something to say, I love to hear from people. So please don't be shy about emailing if, yeah.
Excellent. Well, without any more questions, I think I can wrap it up real quick, and then we can hang out for a little bit if there's anything else. I just want to say again, thank you, Alexa. That was all very excellent information. And thank you all for being here. And before we switch to the next thing, I just want to make sure that you fill out our feedback evaluation. We read every single one, and so that really helps to improve all of our programs. As well, we hope that you get a chance to visit with our vendors at the vendor marketplace and find out about some of the generous discount codes and information from them. Of course, we hope that you head over to our online auction. There's some really fun items to bid on. So check on those items that you are interested in, and make sure that you bid early, bid often, as they say. As well, we have a whole week. Every evening this week, there are more events. So please check your program guide and refresh it to make sure you have all of the most up-to-date links. And again, thank you all for attending this workshop. From all of us at NOFA, we look forward to seeing you next time.
Thank you.
Thank you. This was really great. I appreciate it. I learned a lot, and there's a lot to cover. You covered so much, and it's even though I know some of it, it just, it's like, wow.
Yeah, thank you. Honestly, putting it together, it was hard to figure out what I should focus on. The more I kept thinking about it, I was like, oh, and there's this, and there's this, and there's this. So I'm glad that, I'm glad that you enjoyed it. It's something that I've been learning a lot about, and I'm becoming more and more interested in, and I can tell it's a long learning journey ahead.
Yeah, I'm sure. What would you say in terms of if people want to do more regenerative agriculture, what they can do in terms of helping their soils?
That's a good question. And I think I'm definitely biased because of the work that I do. But one thing that I think really makes a difference is to always have living plants in the ground. So cover cropping, even if it just feels like, oh, if you feel like it's too late, or, there's not enough time, or this or that, it's always good to have something in the ground. The living roots of plants are really doing a lot. You can be saving a lot of nutrients. So I think that's really important. I think also as little disturbance as you can really does make a difference. So I know there's challenges with no-till farming in terms of, I know nutrient cycling works a little bit differently in those systems. And it might take a little bit of time to really get to, because you have more residues to manage and there's a lot of complications, but I think that no-till farming really does help with soil structure, and it really makes a big difference. And then also crop rotation. So just from like a pathology perspective, really getting different crops into different parts of your field at different times is really not only going to support a more diverse microbial population, but it'll also prevent the proliferation of plant pathogens. If you have the same kind of food source or, host in one area at, all the time, then the pathogens are going to flock to that area, and then they're really hard to get rid of. It can take years and years. Some of these fungi have survival structures that can survive in the soil for up to 10 years. So I think crop rotation is something that is sometimes overlooked, but is really important.
So if you're going to have a cover crop, then don't have something that follows it that has the same feeding as the, if you're looking for predators or whatever in terms of the nematodes, or like if you do have a problem with something, either it does something that's out of balance, then you don't want to continue making it worse by something that's gonna, like the cover crop that's the same kind of, whether it's nitrogen fixing and then another legume or something afterwards, like soybeans or something after something else.
Yeah, exactly. Or like brassicas are another one. If you use like a brassica cover crop, you might not want to plant a brassica in the same field. And it's not just with cover crops. I mean, it's also with other crops as well. I mean, crops have very specific, there's some, pests and pathogens that have wide host ranges, but there's some that are pretty specific. And so you don't want to plant the same crop in the same soil like over and over again. Also, they use nutrients differently. So if you have something that's a really heavy feeder and it's going to use a lot of the nitrogen or like a lot of the potassium or something in a soil, you might not want to follow up with something that needs a lot of those same nutrients. I mean, obviously all plants need nitrogen, but that basically they're not heavy feeders, because there's not going to be enough nutrients in that soil, and you're not really going to be building it up in time for the next plant. One thing that our lab does that I think is kind of cool and important is we try to build the soil health and build these nutrients in the soil during production. So like for the garlic project, normally you wouldn't have anything but garlic growing from October to July. And so that's depleting nutrients because it's actively feeding on, it's a really heavy nitrogen feeder, needs a lot of potassium, a lot of phosphorus. So if we're able to kind of mitigate how much nutrients the garlic is taking out of the field by planting cover crops, that'll return some of those nutrients to the soil, or that might fix some nitrogen. That could be a way to build soil during production, which is important. Also, if you have small acreage, you might not have time to always be rebuilding your soils between every single crop. So that's something I'm personally interested in, I think has a lot of potential.
That makes really a lot of sense. And I know people do intercropping with different vegetables and different crops. So that makes a lot of sense, because you're feeding the biodiversity in the soil with the different crops and stuff.
Yeah.
Thank you.
Oh, I see your question. Okay. Am I rotating my fields during my research trials? This is actually, I'm not, and there's a reason for it, and there's pros and cons. So the cons are that obviously I would like to rotate fields, and because of all the things I just mentioned, also because garlic is susceptible to things like wireworm and white rot, and I'm very nervous about the latter because there's a farmer on our road that has white rot in his fields. But The reason that I'm not is because a part of my research that I didn't talk about today is I'm also working on arbuscular mycorrhizal fungi, which is known to associate closely with garlic. And one of the things I'm looking at is if you use an inoculant, because it's, even though it's there's a lot of native mycorrhizal strains, in generally in like conventionally tilled agriculture, you don't see a lot of spores because they're really sensitive to disturbance. They really don't like nitrogen, synthetic nitrogen applications, and they really don't like tillage. So what I'm looking at is if I apply the inoculant over the course of 2 years, can I build up the population enough to actually get colonization and to get some benefits to the garlic. And so that's the reason that I'm not rotating, is because I'm also looking at these field effects of reapplication. But I didn't talk about that today because I think it just gets a little too complicated and it could probably take me another hour.
Thanks. That's really interesting though.
Thank you.
So you're using arbuscular mycorrhizae for garlic?
Yeah, so I'm using a 4-species blend, but they're all in the Glomus genus. And so it's just a commercially available product. And the idea is to look at basically, do these commercial products, can they be useful in agricultural systems? In the second year of the experiment, I'm gonna work with 2 no-till farms and 1 conventional farm. So I'm also interested to see if is it more effective to apply these inoculants in these no-till systems where you have these more structured food webs and you have minimal disturbance and it's already sort of established, or is it not? But yeah, it's arbuscular mycorrhizal fungi, so it'll form arbuscules within the roots that'll serve to exchange nutrients.
Cool. Thank you very much.
Thank you.
I was wondering if you have studied or know folks who are doing similar studies to yours that also include animal rotations in their fields.
That's a good question. And off the top of my head, I don't, but I'm sure that there are those studies going on, maybe even at UMass, but for sure in the Northeast. Because yeah, that's really interesting. And I can actually get back to you on that if you're interested. I'm sure I could ask some lab members who are focused more on grazing. And I think that they would probably know.
Yeah, we were out at Simple Gifts Farm not too long ago. Well, May, so a while ago. But you know, it was like a really inspiring image there because they have got rotation. I think it's 3 years in vegetable production, then 2 years in pasture, or some near there. I thought, wow, if you've got the space and the capacity, that must be like a real boon to, you know, biodiversity in the soil.
Yeah, I was lucky enough to volunteer at a farm in Maine many years, almost 10 years ago now. But they had a really good system too, where they had this like kind of like rotational grazing, where they had cover crops, and then they would mow them down, and they would have their chickens come in, and then they would have their cows come in. And it really was amazing to watch it in action, because that was really the only time that I've seen someone really practicing something like that on their farm and like it really working, you know. But yeah, I should definitely, I'd love to visit Simple Gifts. It's right there. I'm at UMass almost every day, so I'll definitely go and pay them a visit if they'll have me.
I'm sure.
That system.
Yeah. Well, I think some of us are hanging out, so feel free to run. Feel free to hang out. Thanks for joining us.
Yeah, thank you so much. It's been really fun.
Yeah.
All right, I'm gonna take off, but I'll see you soon. And thank you so much for putting this together. It's such a great conference, and I'm so excited about it every year. So thank you.
Thank you.
Bye.
Bye.
Thank you.
Bye. Thank you.